I'm Chris Burrell from the University of Adelaide, and I'd like to welcome you to the Festival of Ideas and to this morning's first session on bats, birds and bugs and us. I guess in the 1960s there were some overconfident medical authorities suggesting infectious diseases were a thing of the past. Technology had conquered at least one of the scourges of mankind. But very soon after that, our complacency took a number of shocks, and hopefully we gained a bit more wisdom from that time. We've seen the emergence of HIV, increasing viral hepatitis, new strains of influenza, many other names to scare the public and to challenge the expert. In one of the most extraordinary and unexpected reservoirs of some strange infections, fruit bats. Now, our speaker this morning is one of Australia's most distinguished medical scientists, veterinary scientists. It's Peter Doherty, who did his veterinary degree in Queensland, his PhD in immunology in Edinburgh, and then returned to the John Curtin School in Canberra, where, together with Rolf Zinkernagel and influenced by Bob Blandon, he made his fundamental discovery of how the T cells of the immune system recognise foreign antigens. A fundamental concept that underpins a lot of our medical science, and particularly immunology. Peter has received many honours and honorary degrees, but I'll mention only two. As you know, he was awarded the Nobel Prize in 1996, the first veterinary graduate to receive this honour, and he was Australian of the Year in the following year. He's currently on the staff of the Department of Immunology at St Jude's Research Hospital in Memphis, and also in Melbourne in the Department of Microbiology and Immunology there, and spends about 3 months of the year in the USA and 9 months in Melbourne. And he's here today to take us through a range of scary, recent, emerging infections, and hopefully to increase our understanding and our knowledge about how these all work. So if I could hand over to you, Peter. Thanks.
Well, thanks very much, Chris. It's a pleasure to be here, and thanks for coming out this morning. It's such a nice day to be outside, isn't it? So I'm still active in science and work mainly on influenza, in fact, but I'm going to review a somewhat broader spectrum of infectious diseases, and I'm aiming to finish reasonably early, and I hope you'll have some questions and we can have some discussion, because I'm not going to say quite so much about remediation and where we're going and why things are getting better. I'm going to talk more about the infections themselves. I've just been in the lab in Memphis, and before I left, I had to go and see the movie Contagion. This is a new movie that's just out, and I was with some trepidation, though the people in the lab had all seen it. They went out for dinner, had some drinks, and went to see the movie, and they said it was quite good. And that's kind of surprising, because Hollywood's treatment of things like infectious disease is usually appalling. I don't know whether you ever saw the movie Outbreak, but it's one of the most abysmal movies ever made. But this is actually quite good, though it does postulate an outbreak or a pandemic which would be much more horrific than anything we've ever seen in our lifetimes or our grandparents' lifetimes. This is an infection that strikes with enormous speed, and it causes a mortality rate of 25 to 30%. This is truly horrific. If you know about the 1918-19 influenza pandemic, which killed many more people in the First World War, it killed about 1% of the human population. This was killing 25 to 30%. That flu was killing something like 2 to 20% of the people who were infected with it. It's worth seeing, I think, and if you're interested in this stuff, and I guess the fact you've come this morning means you are, it's a bit distressing in some senses. Those of us who remember Gwyneth Paltrow as a rather cute Emma Woodhouse, we do get to see the top of her skull taken off, so that's a bit distressing. And it's realistic, because the key players in it are the Centre for Communicable Diseases in Atlanta, Georgia, which is one of the main foci in the world for really handling a novel infectious disease. They operate widely, way outside the United States, and there are somewhat similar situations, setups in Europe, but not quite as substantial as CDC, and the World Health Organization based in Geneva, which monitors global infectious disease and generally does a pretty good job. You know, we don't always think of international agencies as doing a great job, but this one does. Our own preparedness for handling a horrific infectious disease, we have the wonderful veterinary laboratory at Geelong, the Australian Animal Health Laboratory, built to extremely high security standards. We can, for instance, handle the most dangerous pathogen in an animal as big as a horse, which is really a very substantial thing to do. And this is the lab that's done all the work on the Hendra viruses, the Nipah virus. There's a group of about 30 people there working on bat infections. We're building a new microbiology institute in Melbourne. Won't be anything on the level of CDC, but we're bringing all the practical as well as the academic side of infectious disease together, because too often these have become really quite separated, and we're hoping we'll get a much better institution for actually looking at infectious disease in the community. Now, the scenario in contagion is that a virus comes out of a bat, it gets into pigs, it's a paramyxovirus, these are not uncommon viruses, the Nipah virus is somewhere in that sort of category. It gets into a kitchen, the chef prepares the food, people get infected, they all go from Hong Kong, Macau on jumbo jets, and that really rapidly spreads it around the world. And that's a pretty realistic scenario. It's just we haven't seen this sort of 25 to 30% mortality. The reason these types of infectious diseases get around so quickly and are now so much more dangerous in some respects than in the past is, of course, the rapidity of air travel and the fact that people can travel before they feel sick, and also the enormous population size we've achieved. The human population size has more than doubled in my lifetime. We now have almost 7 billion people on the planet. The last time we saw something where 20 to 30% of people were being killed by an infection was way back in the Middle Ages. The Black Death in England, the plague, Yersinia pestis, was killing people at that kind of level. There's been a lot of debate about whether it really was plague, but now people have gone back, they've gone to graveyards from that time, and looking at ancient DNA, which we can now do, they've shown that these people did die from Yersinia pestis. So it was plague that killed them. Plague exists in a rat flea life cycle, and if the fleas get onto humans, they can bite humans and transmit it to humans. The people in London at the time of the London plague in the 1650 or so were killing all the cats and the dogs, which I thought was a really bad idea because the cats and the dogs would clean up the rats. But on the other hand, if the cats and dogs clean up the rats, the fleas look for an alternative host, and that's us. So it might have been fairly neutral, really, whether it was a good thing to do or not. Now, we didn't understand that infection for hundreds of years. It raged in Europe for hundreds of years. And we first understood the germ theory of infectious disease with the work of people like Louis Pasteur in France, Robert Koch in Germany, in the mid-19th century. So it's only 150 years ago that we really began to understand infectious disease, and subsequent to that we started to get vaccines and antibiotics and all the things we're so familiar with today. So it's very, very recent in human history that we actually can understand and handle these things with some sort of speed and effectiveness. Now, bats. Bats have recently come into the scene for infectious disease. If you pick up this morning's Australian, for instance, you'll find there's a whole section in the glossy magazine about bats and how there are too many fruit bats around in some places. We've long known that rabies viruses are carried by bats, by hematophagous bats, the ones that are blood-sucking, and this has been particularly a problem in cattle in South America. And bats have had a sort of pretty bad press from that. There's even some speculation that the Dracula legend relates to people getting rabies and bats, and if you develop rabies for a time, there can be heightened sexual activity, and that could feed into the whole Dracula story. They were more likely progenitor for Count Dracula is actually Vlad the Impaler, which I always think is a very impressive name. Also, the lyssaviruses, these are the viruses that are carried by, that's the rabies type of virus. We've had a couple of deaths in Australia from bat lyssaviruses. So these viruses, not rabies, because it's never transmitted into dogs, and rabies is usually in a dog-human life cycle if it involves us. These viruses are in bats in Australia, or related viruses. And so if you're ever handling a bat, you need to be very, very careful. Though we don't have blood-sucking bats, we do have the fruit bats, of course, and fruit bats have powerful mouth parts to bite through the rind of tropical fruit. So you have to be very careful. It's not a good thing to handle a sick bat. It's a job for bat handlers, professional bat handlers. I don't know how big a profession that is in Australia or what the productivity is in that particular profession, but at least one of the people who've died from these bat lyssaviruses has been a bat handler, and so it's a kind of high-risk occupation. The viruses that are carried in bats. In fruit bats, if you'd asked us 10 years ago, 12 years ago, what viruses there are in bats, we would have only come up with the rabies-type viruses. Now there's a whole list of them. For many years, we were searching for the maintaining host for the virus that causes Ebola hemorrhagic fever and Marburg hemorrhagic fever. These are horrible diseases that kill very rapidly, people bleed, they're quite infectious, and they can infect medical personnel in contact and all the rest of it. We didn't know what was maintaining them for a very long time. Recently, they've isolated Marburg virus from bats, and they've got serological evidence that Ebola virus is in bats. So it's pretty likely that these viruses are carried by fruit bats in Africa. They're not outside Africa at this stage. Though there's a variant, a very mild variant, that caused a disease in monkeys in the US, and it's also in the Philippines. The problem with Ebola and Marburg, or Ebola particularly, is not only does it infect humans, it's also been taking a considerable toll on the chimps and the gorillas and the large apes in Africa. And we really don't want to lose those. And we're close to having a vaccine. It's in advanced testing at the Vaccine Research Center at the National Institutes of Health in Bethesda, Maryland. And we can get that into people, like at-risk medical personnel, but how do you get it into gorillas and chimps? I mean, they're quite friendly, but when you start sticking needles into them and shooting dart guns into them, they're not really so friendly. So the question would be how we actually spread that vaccine around. And then we got SARS, the sudden acute respiratory syndrome that hit in Asia in 2002, 2003. That was a very scary, very rapid onset and quite circumscribed infectious disease outbreak. It started in China, and it took us several months, a couple of months I think, to work out what the actual infection was. People thought initially that it was influenza, perhaps the avian influenza that people have been first encountered back in 1997 with the outbreak in Hong Kong, and quickly found that it wasn't that. But the people in that global influenza network then worked on it and worked out fairly quickly that it's actually a coronavirus. Now, these are viruses that normally cause respiratory infections in us. They're viruses that cause common cold type symptoms. They're not uncommon, they're in all sorts of species. And it turns out that the initial thinking was, or the initial evidence was, that this had got across into humans from a little animal called the Himalayan civet cat. This is a wild living animal. It's not a cat, but it lives in the wild, and it was being sold in live animal markets in China, because they have the habit of eating live animals or taking wild animals, killing them, and then eating them in restaurants and so forth. And the people who handled those animals in these live animal markets were actually found to have antibodies against this, and had antibodies for some time. That means they'd had subclinical infections. The virus itself generally wasn't too severe, didn't cause too severe an infection in young people, but it was highly lethal as people got older. And this is not uncommon with virus infections. As we get older, our immune system fails when we encounter something new particularly. And so older people are generally most susceptible to infections, much more susceptible than young people. In the end analysis, once we understood what the virus was, we were able to work out how it was transmitted because we could measure it, we could find it, and we discovered why it was that a lot of the people who died were actually medical personnel. The reason for that is that this virus, when you get infected with it, we don't push out much virus early in the infection, and we don't actually start to push out a lot of virus until we feel really quite sick. We get into the hospital, and then the medical doctors and so forth got infected. Now, they were thinking it was more like influenza, which is exactly the other way around. You put out a lot of infectious virus early. By the time you feel really sick, you may not be all that infectious. And they treated it like that, and it turned out to be a dangerous way to handle it. So basically, we unlike the plague, which took hundreds of years to work out, we worked out this infection in several months. And we were able to then implement suitable quarantine and procedural changes that meant that it stopped spreading. And the whole thing was over. About 8,000 people in total were infected. About 800 of those died. There's a lot we don't understand about it. We know that there were some super spreaders who carried it firstly to Hong Kong, then one person carried it to Toronto and triggered an outbreak in a hotel in Toronto. Toronto suffered major economic loss as a result. Another single person took it to Singapore, and so on. So there's a lot we don't understand about it, but at this stage, the infection seems to have gone away, though it's still presumably out there. Now, we later found that the civet cats had most likely got the virus from fruit bats, that it's a virus that's in fruit bats, and these coronaviruses, which we didn't know about at all in bats, there are something like 22 of them, in fact, and this is one of them. And so we're now finding that there's this enormous reservoir of virus infection out there in fruit bats. And of course, we're all very familiar in Australia with the story of Hendra virus, a virus that's coming out of fruit bats, infecting horses, then creates a major risk for veterinarians and people who are handling horses. Horses are very big animals. If a horse is incubating a respiratory infection and you wash out the nose of a horse, you're talking about gallons and buckets of stuff. And so it's not like a human infection or something like that. It's a lot of material, so there can be a lot of aerosol around. So people are very at risk. The Hendra situation is, there's been increased numbers of cases in horses, far north of here on the whole. It's killed several people and killed them horribly. It's a worse infection, I'm told, than Ebola, which seemed to be the worst infection in the world. The group at the Geelong laboratory, the Animal Health Laboratory, has isolated the virus. They've made a vaccine. The vaccine is currently being tested and it will be given to horses. Once we start to vaccinate, once we vaccinate all the horses that are in close contact with people against this virus, then we should be able to remove the threat. So far, it hasn't gone directly from bats to humans. There's always the potential of that. I mean, the bats hang upside down in trees. The virologists isolate the virus on plastic sheets just from the droppings under the trees. And there has recently been more virus on those sheets. They think there's been a lot of stress caused by the high rainfalls and so forth that has actually stressed the bats, and they're pushing out more virus. Another virus that also was worked out at the Geelong lab is the Nipah virus. It's a different type of virus. It goes from fruit bats into pigs, and then pigs give it to humans, though there is at least one account of this virus going directly from bats to humans. So these are viruses we have to watch pretty closely. And actually, the virus in the Contagion movie is actually a fiction created by an American virologist called Ian Lipkin, who was an advisor to the movie, which actually combines a lot of the characteristics of Nipah virus and Hendra virus. And in fact, Australia gets a mention because the virus is initially isolated by a courageous virus who grows it out in a bat cell line from Geelong. Of course, we can feel good about that. And we should feel good about the Geelong lab. It does a really good job. And there are lots of other viruses that are kind of on the move. In Australia, we've seen chikungunya virus, the virus that causes epidemic polyarthritis with rash, move right down from the north into the south. We've seen Obama forest virus, a rather similar virus, also emerge as a cause of that sort of infection. In fact, Ross River's also gone across to Perth. in the Asia-Pacific region, and right up into the Middle East and into Italy, in fact, there's another related virus called chikungunya, which has been moving quite fast. And we don't know why. It was around for years, and suddenly it starts to spread, and we suddenly start to get a lot more cases in different places. And people are developing vaccines against that. And these infections are constantly out there, and there's this constant reservoir in wildlife and animals that can jump across into us, and in some cases become established in us. And that's been happening for millennia, but the pace seems to have accelerated, possibly to do with large human population size, erosion of habitat as we take habitat away from wild species, and rapid transport. Here's an infection that got into the United States in 1999. Suddenly in New York City, people noticed that birds are dropping out of the sky, particularly crows. Birds were dying at the Bronx Zoo. And then they started to get cases of encephalitis, neurological disease in humans. And people tied that together, and it turned out that this virus, West Nile virus, which has been around a long time and was causing some disease problems in southern Russia, Israel, and that area of the world, had somehow got across to the United States. Now, this is an insect-borne virus, and which goes in a bird, insect, human or occasional human life cycle. We're not important to it. It's maintained in the bird-insect life cycle. But we occasionally get bitten by an insect. The virus multiplies in the birds. It also multiplies in the insects, as these types of viruses do. And they had a pretty significant outbreak in New York City. And then over the next 5 years, more rapidly, in fact, it spread south, because that's the way the bird migration patterns go. They go north-south more than east-west. But it gradually spread across the United States. It took 5 years to get across the United States to California. And it's now in the northern part of South America, and it's right across the US. It's up into Canada. It almost wiped out the yellow-tailed magpies in California. So how did that virus get in? We have no idea. It could have come in the arm in an infected person. Someone may have been bitten by a mosquito. They may have been in an early stage of infection, circulating quite a lot of virus, maybe feeling a bit off color, and they might have just been bitten by a mosquito. It could have come in an imported bird, either a legally imported bird to the zoo, for instance, or an illegally imported bird, because there's a lot of illegal importation of exotic animal species. And in fact, with exotic animal species, there's often rather little in the way of quarantine. The sort of thing we'd use, say, for horses, we watch very closely. But a lot of exotic animal species we don't watch nearly so closely. There's a vaccine for horses. We don't have a vaccine for humans. It's easier to get a vaccine on the market for horses. And then there's always the question with humans, just how much of an incidence of infection we need before it really becomes economic and medically practicable to put a vaccine out there. But there is a vaccine very close to availability for humans. So we've got this enormous spectrum of infectious entities which can infect us and cause problems. We can't predict what they are. Some of them are quite novel, and they're coming along all the time. And the variety of these things is vast. So we have to have a very, very complex immune system to handle them, and that's what we have. The actual word immunus means without tax. It comes from the Latin, where some of the soldiers who come back from the wars were for a time exempt from tax, like Tea Party Republicans in the United States. And the tax, of course, that the immune system defeats is the tax of infection, that invasion by much simpler life forms that can multiply very quickly and change very quickly by mutation. And it has to deal constantly with this change. It divides up into two bits. One's the innate immune system. That's kind of a first response. It's got levels of specificity, but it's much more a sort of a sudden dumping of stuff, like toxic chemicals and things, out of various cells in an attempt to kill off the invading organism. It's very powerful, and it also helps set up the second part of the immune response, which we call the adaptive response. That's the response that we prime with vaccination, the response that really clears out the infection in the end, and that's the response that has what we call immunological memory, which is the basis of vaccination. You give a bit of the bug, or whatever it is, or a killed form of the bug, or a less lethal form of the bug, we make an immune response, and then we have, in many cases, lifelong immunological memory. yellow fever vaccine, for instance, that some of us will have had, and we will still have if we're going to West Africa, where it's still a problem, will give us lifelong immunological memory against that thing, though the medical people would say you should be boosted at least every 10 years. So the innate immune system is very primitive, actually. It goes right back in evolution to the simplest life forms. The adaptive immune system comes in with actually the bony fishes, and it's in us, and it's in birds, and in reptiles. It's diverged evolutionarily over time. The bird immune system looks rather different from the mammalian immune system, which again looks a bit different from the reptile immune system. But the themes are the same, and the basic mechanisms at the end are the same, and it does the same job, which kind of shows there's more than one way of skinning a cat in evolutionary sense. And I think, in many ways, our immune system's probably or the mammalian one's probably the most effective. When it comes to Nobel Prizes, until this week actually, the only Nobel Prize that had been given for the innate immune system was in 1908 to Eli Metchnikoff for discovering the macrophage, though his pupil Jules Bordet, won the 1919 Nobel Prize for discovering a substance called complement, which sort of helps out the immune response. And the second Nobel Prize for the adaptive immune response and for the theory of adaptive immunity went in the same year to Paul Ehrlich, who's really the father of immunology, and there were no mothers of immunology at this stage. We've been growing a group of mothers of immunology more recently, fortunately. Now this week we heard the announcement of the Nobel Prize for Medicine, and these two guys got it for innate immunity. They got it for working out the details of that early response to infection and how that kind of works in many ways, and very powerful sets of experiments. And then this man, Ralph Steinman, got the Nobel Prize for working out what we call the dendritic cell. This is the cell that's worked on by the innate immune response and sets up to stimulate the adaptive response. Ralph, unfortunately, had pancreatic cancer for 4.5 years, which is almost extraordinary for pancreatic cancer. He'd taken every experimental therapy in sequence, including his own dendritic cell therapy, where they feed killed tumor cells to these dendritic cells and then inject them back in. And he had survived substantially, but he died on the Friday before the prize was announced. The Nobel Prize Committee wasn't aware of that when they announced the prize, and were only informed later. And they decided against their rules, actually, that the prize would stand, which was great. It would have been terrible if it hadn't. Now, the basis of vaccination is immune memory. And basically that means that cells that are stimulated at the time of the initial immune response that are highly specific and have very specific receptors for a particular part of the infectious entity survive in the long term and can be recalled rapidly to immunity. We've known about this in a sort of operational sense since the time of Thucydides and the Peloponnesian Wars. He recognized for the plague in Athens, which was probably typhus, that those who had nursed the sick could not catch the disease a second time, and they were in fact protected. But that protection was specific. It was only for that infection, not for all other infections. That's a very good description of immune memory and immunity. But the next really good descriptions of what was happening really didn't come along until the early 20th century, late 19th century. And we've been working out those immune responses through the 20th century and into this century. Still a lot to understand and a lot of work to be done. The most important component of the immune response as far as vaccination is concerned is the antibody molecule, produced by cells called plasma cells. These are big factory cells that pump out these molecules that circulate in the blood. And we've been finding recently, with better technologies and so forth, that people who've had an infection, say 50 years before, may still be making antibody against those same things that they saw way back then. So immunological memory can be very, very long-lived indeed. And that's what we try and stimulate with vaccination. We have vaccines that work quite well against a number of infections, particularly those infectious agents that don't change very much. The measles vaccine, the polio vaccine, and cause their disease by going through the blood. They have what we call a systemic pathogenesis. That is, the virus multiplies, say, with polio, it multiplies initially in the gut, gets into the blood. You don't even notice if it's multiplying in the gut, gets into the blood, and then if it gets into the large motor nerve nerve cells in the spinal cord and the brain, which it does in a few people, then you get poliomyelitis because the virus kills those cells. So if you can stop it in the blood, which you can do very well with antibodies, you don't have a problem. Same is true of measles. It starts in the oropharynx. It's true of a lot of the insect-borne infections. For instance, the yellow fever, West Nile and so forth, and these vaccines are great. But there are a lot of challenges in vaccinology. We don't have vaccines against HIV/AIDS, and we don't really know how to make one, though we've been trying enormously hard, and we've spent very, very large amounts of money on it. We don't have a vaccine against hepatitis C, which again is a virus that changes very quickly. So there are a lot of challenges out there, and if you're young and you're thinking you might like to do microbiology for a career, don't think you're going to run out of problems. There's going to be lots and lots of things to do, lots and lots of challenges to solve. We have eliminated one human pathogen, one terrible human pathogen, that until several years before the final elimination was completed was still killing tens of millions of people, and that was smallpox. And we eliminated that with a very basic sort of vaccine, a live attenuated vaccine, not that different from the vaccine that Edward Jenner used back in 1796 when he realized that cowpox virus protected against smallpox. And the lead figures in that elimination campaign, the people who really administered it and made it work, were Frank Fenner and D.A. Henderson and Isao Arita. Now, Fenner died last year at the age of 95, and he was an Adelaide boy. He was educated at Theberton High School and at the University of Adelaide, where he did medicine, had a distinguished career early on in the Australian Army in malaria control during the war, then was the first head of microbiology at the Australian National University, which is where we did our work. So he kind of is a grandfather in science, if you like. I don't know whether you're aware of it, but I think that Adelaide actually has more Australian Nobel Prize winners to its credit than any other city. It has Lawrence Bragg, who shared the 1915 Nobel Prize with his father. It has Howard Florey, who shared the Nobel Prize for penicillin. And it has Robin Warren, who shared the Nobel Prize with Barry Marshall for Helicobacter and stomach cancer. The second virus to ever be eliminated from the planet was eliminated this year, and that's a veterinary virus. It's the virus that causes rinderpest in cattle. This was a devastating disease that caused enormous economic loss in Britain and Europe through the 19th century. It really led to the rise of the veterinary profession in some ways, the attempts to deal with it, and it was still causing very large numbers of deaths in cattle in Africa up into the 1980s. It was eliminated by a vaccination campaign, not a particularly sophisticated vaccine, one that was rather like the polio vaccine and the smallpox vaccine. The elimination, it was announced officially this June that rinderpest had actually gone, which is really a major triumph. Now, rinderpest is a virus that diverged from human measles, and rinderpest are thought to have diverged about 1,200 years ago. So it's very closely related to human measles. We've got rid of rinderpest. The challenge is to get rid of human measles. One of the problems with getting rid of human measles is convincing everyone to vaccinate. And that's been an issue, of course, this anti-vaccination sentiment in the wider community. Also, measles is incredibly, as is rinderpest, incredibly infectious. Influenza. I'm going to finish this with talking about influenza. Influenza is an infection we're never going to eliminate. And the reason for that is it's basically an infection of wild birds. It's an infection of aquatic birds. And there are tremendous numbers of variety of influenza viruses in the wild birds. There's no way we're going to kill out all the wild birds, so we're going to be stuck with influenza forever. That was kind of discovered, that influenza is basically maintained in nature by wild birds, back in the 1970s. But in 1918-19, when there was the terrible influenza pandemic, there was this little rhyme. I had a little bird and its name was Enza. I opened the window and influenza. I think those of us who are older probably remember that, but I think younger kids don't seem to know it anymore. But it was kind of perceptive that the bird influenza tie-up really preceded the science. Influenza virus, it's a virus that changes a lot, it mutates. As you know, we have to get a new vaccine every year or so because of the mutations. It's got no proofreading really that matters. And there are two surface molecules, the hemagglutinin and the neuraminidase molecule, that attach to the cell and get the virus into the cell, or the neuraminidase helps it get away from the cell. Most of our effective antibody response that protects us against us are directed at these hemagglutinin molecules, and the antiviral drugs Relenza and Tamiflu are actually directed against this molecule, the neuraminidase. What they do is they stop the virus getting away from the cell. So new virus is made. If it can't get away from the cell, it can't infect more cells and the infection doesn't go ahead. So these antiviral drugs give us a breathing space while the adaptive immune response takes over and can eliminate the virus. And if you give them early, they really work pretty well, though there are some mutants that escape these viruses, these drugs, like Relenza and Tamiflu. This shows the full range of influenza viruses that are in birds, and this shows the species they jump across into. The H3 is, the ones in humans are H1, H2, and H3. H2 is not around at the moment. It caused a big outbreak in 1956 and caused a number of deaths. H1 is the H1N1s are the ones that are causing issues recently. H3N2, the Hong Kong flu, has been around in various guises since 1968. We catch influenza viruses from other human beings. We don't normally catch them from birds. H3, H5, H7, and H9 are the only ones that are known to come across to us from chickens. Others come across from ducks, for instance, and they may mix in a pig before they infect us. The pig respiratory tract is very like ours, and it can be infected by both avian and human viruses. They can mix up there because the flu virus genome is in 8 different bits, and it can sort of mix up if you get a cell simultaneously infected with 2 different flus. H5N1, the bird flu that we all worried about so much a few years back, first came out as a highly virulent or high pathogenicity virus in chickens in 1997. It circulated for many years in chickens as a not very virulent virus, but then suddenly it started to kill lots of chickens, and it also infected humans. And these were people who were getting the virus directly from the birds. Hong Kong, China, has a culture of live bird markets. There are none in Hong Kong now, I think, but they have live bird markets. People like to get a live chicken, then kill it themselves for cooking. And this was the source of infection, the live bird markets. They had to kill enormous numbers of birds. That virus now has been on the move since 1997. It changed in about 2005 and spread right westward into Europe and North Africa, killing large numbers of birds, and large numbers of other birds had to be culled to try and control the infection, and some humans. So far there have been about 600 people infected with this virus, with about 60% mortality. Latest mortalities just this year, 8 people got infected in Cambodia and 8 people died. So that's not very comforting. Recent experiments that I just heard about at a meeting in Europe were that the Dutch actually took this virus, they passaged it serially through ferrets. That's ferret to ferret to ferret transmission under very high security conditions, of course. Ferrets have a disease that's very like our disease, and they showed that 5 mutations in the virus would change it to a virus that would transmit between humans, which was a big concern. It could change, and it's being watched very closely by World Health Organization and the various influenza labs. 1918-19, unusual in that the disease killed young people particularly. Of course, we couldn't isolate influenza in 1918-19. It's thought that the fact that it killed young people reflected an over-response on the part of that innate response, that it was flooding the body with these toxic chemicals, trying to kill off the virus, and that caused vascular leakage, and people essentially drowned in their own lung fluids. We saw a bit of that. We see that also with the H5N1 virus in humans. Those occasional cases in humans, we get this very, very severe early pathology, and the thought that it's what we call a cytokine storm or a chemokine shock that happens very early in the infection that actually causes acute death. Now, we don't understand a lot of the details of that, and we do need to understand the details, because if we get that type of effect, we need to know what parts of that cascade we need to target with drugs and therapy. So there's a lot of work going on now on trying to understand those early events in influenza infection, and that's what our Memphis lab's actually involved in. The virus wasn't the first, that was 1918-19. First influenza virus wasn't isolated from humans until 1933. They got it out of pigs a couple of years earlier, and they reconstructed that 1918 virus using the PCR technique, which of course is again a more recent innovation, the one that they use to identify rapists and all that sort of thing. And they know it's got all the characteristics of a highly virulent virus, largely a virus of bird origin, though it's not known exactly where it came from. They got it out of fixed tissue material in a pathology lab in Washington, and by digging up people who were quickly frozen in the permafrost up in Alaska. And actually put the virus together. It's a terrible virus. It's been put into monkeys, causes rapid and horrific disease, and is just as bad as the one that's in that Contagion movie. Flu, so infectious because we are infected before we feel ill. We cough and splutter the virus. We get on planes, we travel, and here's it going across. This is a seasonal influenza going across the United States in 6 weeks. And the H1N1, the pandemic flu that we've just had, went round the world in less than a year. You can see the red is actually flu. It's not Republican. And like a lot of bad things, it started in Texas and it went right across the states in very short order. Pigs mix up these viruses. I won't go into the detail. The swine flu that we all encountered in 2009 happened when 2 pig viruses got together. The North American swine virus had changed quite a bit over the last few years, and it was what we call a triple reassortant. It had bits of viruses which are characteristic of those that affect humans, that infect pigs, and then infect birds. So we'd got quite a mix in this pig virus. Usually the pig viruses don't change very much, but this virus has started to change a lot, and we don't quite understand why. We think it's because of one of the genes that have come from the chicken. And then it got together somehow with a Eurasian pig virus. Now, how a Eurasian pig virus got together with a North American pig virus, I don't know. And pigs don't normally take package holidays to Mexico, but it did, and someone brought across a pig that was infected. They mixed up again, and they gave us a virus which grows very rapidly in humans. It is also what we call a stealth virus. It doesn't turn on that innate immune response quickly, and that's why this virus spread with enormous speed. Now, when that infection kicked off, it kicked off in Mexico, and there were a lot of deaths, and people were very worried because they thought this is an extremely severe infection. Turned out, as time went by, we realized it wasn't that severe. It was severe in some groups. It put 6 times as many indigenous people in hospital all around the world. It put 6 times as many pregnant women in hospital. It particularly infected young people, but it didn't so much affect older people. If we were born before 1950, we seemed to have some cross-reactive antibody from an earlier virus that protected us. So we didn't see that usual death rate in older people. It used up all the critical care beds pretty much. It used up all the ECMO machines, the heart-lung type machines. They kept a number of people alive by perfusing their blood and allowing the machine to breathe for them until they actually got through the crisis and the immune response got rid of the virus. So it's actually quite a bad virus in some senses, but in general, most people, it wasn't too severe. This was the British outbreak in 2009, and I show that because it's very interesting. You've got this peak of cases of people being admitted to hospital, then you get a big trough, and then you get another peak. This is school vacation. You get the kids away from school, you slow down the pandemic, and it's the kids that are bringing it home into the families. But then you can get a problem. I mean, if you send all the kids home from school, and the parents work out that they can't take time off work, so they get all the kids together in one house with one parent looking after them, you've got exactly the same effect. But this was the predicted school holiday, so people were prepared for it. Antiviral drugs I mentioned. We're doing a lot on new vaccines. There's a whole lot of research going on trying to make more cross-reactive flu vaccines and to trying to make one which we could prepare ahead of time. Because the issue with the H1N1 flu, it got around the world in 6 months. And it was first detected in the US in April. It was in Melbourne in May. In fact, Melbourne was given separate status as a major pandemic centre. It was sort of separated from the rest of Australia, which of course some people would probably want to do. And it was in Hong Kong in May as well, so it really got around very quickly. So there's a whole lot of research going on here, a whole lot of exciting things happening in vaccine research, and we hope to have much better vaccines and much more cross-reactive vaccines over the next few years. And basically, as far as pandemic threats with flu go, we're not quite so worried about the H5N1, though we're watching it. But these viruses are actually reassorting in pigs at a very high rate. And we just saw another one come out into humans, but it didn't cause much infection and infected a couple of kids and hasn't really spread. But we're watching these very closely because something has changed in the pigs, and these are probably the main risk factors for us. Okay, thanks. That's it.
Thanks very much, Peter, for that very stimulating, informative overview. We've time for questions. Is there anyone who has a question from the audience? There's a couple of microphones there. I think there's one just in the centre. And if anyone has a question.
With the bat viruses, what's the sort of evolutionary advantage that they've cohabited with these viruses for so long and then suddenly appeared? And are there other groups of animals that are potentially harbouring similar nasty viruses?
Nobody's really much looked at bats, and so we know nothing about their immune system. And the group in Geelong now has about 30 people on bats and bat immune systems. So we don't know what it is that allows these viruses to persist in this species. I can't believe there's any evolutionary advantage for the bats. But we have to think it's something, the fact that we're suddenly starting to see them more in humans has to do, I think, with erosion of habitat, but also bat numbers in some places seem to have increased greatly, though bat numbers, say, in North America are very threatened by the infection of bats. So what exactly is happening, we don't really understand. And it needs a reasonable amount of research, but it really hasn't it's received no attention over the years. Other viruses that are out there, we know of various rodent viruses that occasionally jump across. For instance, Korean hemorrhagic fever was discovered during the Korean War when you had a lot of young Americans and Commonwealth soldiers out sort of camping in the environment, and this Korean hemorrhagic fever virus came across from a small rodent species, Epidemus, and infected them. And so suddenly we saw that. Then in southern United States, when people started to be maybe more adventurous in their outdoor experience and so forth, we saw a similar virus called Sinombre come across into humans. So the more humans come into contact with wildlife species, the more of these things we know. So otherwise we don't know of vast reservoirs, but who knows?
Hello, Peter. I've associated flu viruses with the winter months. Is that not the case?
It tends to be in Australia and the UK, but Hong Kong gets 2 distinct flu seasons a year, and my perception of Hong Kong is the difference between winter and summer is really quite insubstantial. But generally, flu in our societies is much more a winter disease. Yeah, the Brits. I showed those 2009 peaks. Early on, they were giving a lot of the antiviral drug that they'd stockpiled for the possible H5N1 flu. So they used a lot of that drug. It works against any flu virus, including the flu Bs. Used a lot of it early on, then decided the disease wasn't so severe, so they backed off on it. Then the 2010 winter outbreak was actually resulted in a lot more hospitalisations, and they actually think that may have been related to the fact they weren't using the drug as a prophylactic. But it does tend to be a winter disease, yeah.
Can we control the numbers of fruit bats? Do they aid in pollination?
I believe they do aid in pollination, and they presumably aid in insect control. But I think we have to be, you know, we have a kind of a thing that's referred to in the United States as the Bambi syndrome. You know, a lot of communities and suburban communities are kind of overrun with deer, and as a result of that, people get Lyme disease because of the life cycle of that parasite. But there's a tremendous reluctance to do any culling because everyone's, since Hollywood sort of created Bambi and Mickey Mouse and so we all anthropomorphize these animals, and so it really needs some culling, but there's just a lot of social pressure against it. Whether that's true with the bats or not, I don't know, but I think that's why science is so important, because what we need is our epidemiologists and our people who study bats out there analysing bat numbers, where they are, what they're doing. And if bat numbers are totally overwhelming cities and bat numbers are increasingly massively, maybe there is a case for culling bats in some situations. I think what we've come to, with 7 billion people on the planet, I believe we have to manage the planet. And we have to manage other species for their survival, and we have to manage the planet for our survival as well. And that's, you know, not a particularly popular view, but I think it is essential.
People have a lot of contact with cats and dogs, but they haven't really been mentioned. Why is that?
Well, we know what we do get from cats. We get cat scratch fever. I mean, Chris is a medical doctor, he knows this stuff. We don't usually get much from cats. Cats were very susceptible to the H5N1 bird flu, and they fed a lot of carcasses from these birds to the leopards and so forth in, I think, the zoo in, I think it may have been Vietnam or somewhere, but they fed a lot of zoo animals, and a lot of the big cats died. So cats were really quite readily infected with them. We worried about the fact that cats might spread them, but we haven't had a problem with that. The dogs, until when I was a veterinary student years ago, I don't think we knew about influenza in dogs at all. It wasn't a problem. But influenza jumped probably from horses into dogs in the United States some years back, initially into greyhounds, then into dogs in general, and dogs are now vaccinated against it, I think. But I don't know of any situation where humans caught an influenza virus from a dog. The thing we worried about with dogs, of course, was hydatid disease. But that's a parasite. We've had big control measures. You need to keep dogs away from sheep and all sorts of things. And I think that's pretty well controlled in Australia and New Zealand, for instance.
How do you think, is there any relationship between outbreak of genetic modified food production and appearance of new viruses?
No, there's nothing that would link anything that I know about with GM food to viruses. I mean, these are not viruses that go into plants at all. We're not doing any genetically modified animals in commercial operations. There's really nothing that would relate the two, and none of those genes that have been put into plants for various reasons emerge in the viruses or the infectious agents. You know, the way they isolate these viruses now, or analyse them with flu viruses, they do what's called deep sequencing. They'll sequence the whole viral genome. We know exactly what's there and where it's come from, and there's no hint that any of that would happen, and it's kind of unlikely, actually, that it would happen. Yeah. By the way, we pulled out SARS in a couple of months. In the Contagion movie, they show them diagnosing it really in a few days, and that actually is quite realistic now. We now have chips which would have all the virus genomes on them, including some low stringency copies where it's a related but unknown virus would probably be picked up. We probably get to know what a new virus is that's causing a major outbreak very, very fast indeed.
Do we know why aquatic birds are such prolific carriers of viruses?
Yes, I think so. This is actually a perfect ecological system for maintaining a virus. The virus infection, influenza infection in birds, aquatic birds, is normally a gastrointestinal tract infection, not a respiratory tract infection. The high pathogenicity ones that kill wild birds, as happened with the H5N1 when it mutated will kill wild birds, but normally it doesn't. So you've got a virus which is a bit like the bat viruses, that infects the birds, doesn't cause them much problem, and it's excreted into water. Influenza viruses survive very well in water. And so what you've got is, and if you have for instance, as happened, say, in Qinghai Lake in Western China, you have a whole lot of different species of aquatic birds all coming together on this lake. Now those birds, different species, they're genetically quite different, and even if one is susceptible, as the geese were, to the mutated H5N1 virus, and it kills the geese, it didn't kill the ducks. So the ducks didn't have a particularly bad problem. So they were the ones who carried it westwards. So having some sort of common source of infection with a whole lot of different species with different levels of disease is a perfect system for keeping any sort of infection going. The other thing that factors into it too is that chicken waste is often flooded onto rice paddies. And again, the ducks come onto the rice paddies. So again, the agricultural practices in that part of the world are part of the threat. So it's a very interesting study for ecology and epidemiology, in fact, though of course, you know, we get money for studying it so we can protect humans and domestic animals. But, you know, the fact that 1 billion chickens have been killed as culled or killed by the infection with H5N1 represent tremendous economic loss in the developing world, and a lot of expectations having to be lowered as a result. So these are, even if they're not infecting humans in a big way, they can be an enormous human problem.
Thanks. Well, it'll be good to carry on, but we've got instructions to finish on time. So I think this has been a most illuminating session. Peter, I'd like to thank you very much for steering us through this so well, so eloquently, and to thank the audience for coming and your participation. Thank you very much.